Welcome everybody to our new video, and in this video we will talk about pairing of nitrogen bases. But before I'll start with the video, I want to give you a little introduction. I'm from BBLC, that is Brain Based Learning Center, and on this platform we are bringing the biology classes for +1 and +2 students. So if you are preparing for your board exams or your entrance exam, you can take the benefits of this video. And if you want to enroll for the full course, there is a link present on our social media. You can click on that link and enroll yourself for the full course. So this is about the BBLC. Now let's begin with the topic, that is pairing of nitrogen bases. This is a very simple and short topic which we're gonna study. So let's move on. So now we are going to talk about pairing of nitrogen bases. So in previous videos related to DNA, what we have talked about, first we talk about the monomer of DNA, that is nucleotide. Then we talk about how to join nucleotide, right? Then we talk about DNA double helix structure, right? So from these three topics, I believe you understand regarding the DNA structure. So what is a DNA structure? We know that DNA is a double chain structure, and in this double chain there is a nitrogen base pairing, right? So how the base pairs attach? Like this. So two base pairs are attached in a DNA double ring. So in this video, we are going to talk about how this base will pair up in a in a DNA chain. Right? So we all know there are two nitrogen bases, and this is how they are attached. So now let me talk about the nitrogen bases. There are two type of nitrogen bases. We know about this. First is a purines and second is a pyrimidine. We know that purines are double ring structured carbon nitrogen structure, double ring, right? And the first one is known as adenine, and second one is known as guanine, right? And then we have pyrimidine. In pyrimidine, we have one ring of carbon nitrogen. So this is a pyrimidine. So in pyrimidine, we have thymine, and then we have the other is cytosine. So let me make the cytosine, and next we have cytosine, right? So in DNA structure, what we have thymine, right? But in case of RNA, we have another pyramid which is known as uracil. So we don't need here because we are talking about DNA structure. but I'm just making a uracil structure just to make you remember that this is one of the pyrimidine which is present in RNA. So this is another pyrimidine which is present in RNA, and it is known as uracil, only in RNA, right? Okay, so just let me color this. I just want to make a, no, I want to distinguish pyrimidine from purines. That's why I'm coloring this. All right, so this is a purine, and another one we have pyrimidine. So this is, we have pyrimidine, right? So now our topic is how these nitrogen bases are attached. Okay. Uracil, I'm coloring it with different color. Just want you guys to remember that this is a part of a RNA. Okay. So now remember one thing. This is adenine, this is first purine, and then we have thymine. So what do you think? So any adenine with will go and make a bond with any pyrimidine? What I'm trying to say, what do you think? Any purine will make a bond with any pyrimidine? Is it so? No, it's not like this. Always remember, adenine will make a bond with thymine, right? Adenine will always make a bond with thymine, and guanine will always make a bond with cytosine, right? So let's make this, join this. So what I'm saying is adenine, I'm writing just A just to make you understand, not making a full structure. Adenine, that is a purine, will always make a bond with thymine, right? It will not make bond with any other pyrimidine. You can say that is a cytosine. It will not, and it will make a bond, and this bond is known as hydrogen bond, right? So this bond is known as hydrogen bond, and it will make two bonds, right? Adenine and thymine will make two bonds. So next pyrimidine we have guanine, and guanine will always bond with cytosine, and it will make, and the bond between guanine and cytosine is again hydrogen bond, and we call it H bond, right? And it will make three bonds, right? Guanine will make three bonds with cytosine. So always remember, this is a case of DNA. Now in case of RNA, what will happen? A, that is adenine, will make a bond with uracil, not with thymine. So thymine is replaced with uracil, right? So this is a case of RNA. Now next thing, I hope this much is clear to you, that A will make a bond with T and G will make a bond with C, right? Now I want to ask you something. We are saying that DNA is a double chain structure, and nitrogen bases are there in between. So what do you say? Is it on one side all the pyrimidines are present, and on the other side all the pyrimidines are present, and then they are making bond? Is it so? No, it's not like this. So this is not right. So what will happen in one chain if nitrogen bases are present? So in this chain how the nitrogen bases are present. So this is a pyrimidine, sorry, purine. Then we have a pyrimidine. Then we have a pyrimidine. We can say then we have a pyrimidine, and then we have purine. So this can be anything, right? On a chain, any type of purines are present, any number of pyrimidines are present. But for one species, it will remain same. For example, in you can say in cat, it is different, but it is a one species. So in one species, it is same. For human beings, it is same. So these are, this could be anything, pyrimidines, purines, anything. But what is important, that purines will always make a pair with pyrimidine, right? So A will always pair with T and G will always pair with C in case of DNA. So now we can make the another chain. So here we have purine. So on the other side what we'll have, that is pyrimidine, right? So here we have pyrimidine, now we have purine. Pyrimidine, now we have purine. So now we have pyrimidine, now here we have purine. So this way, here we have purine, now we have pyrimidine, right? So purines will always bond with pyrimidine, but it's not like this that on one side all purines will present and on the other side all pyrimidine will present. No. So purines, pyrimidine, pyrimidine, purines, anything can be possible. But what is to remember, that always purines will make a bond with pyrimidine. I hope this is clear to you, right? Okay. Now we need to talk about the next thing, that is Chargaff rule, right? So what we are going to talk about, that is Chargaff rule. In that Chargaff rule, we're going to talk about how these, what is, what are the rules of these pyrimidine and purines. We need to talk about that. So let's talk about Chargaff rule.